All right, this is part two of three parts in chapter three, and we were talking about proteins, the fact that they're built out of amino acid monomers, and we form this long polypeptide chain. And I mentioned that for that protein to really be functional biologically or chemically speaking, that chain has to be folded up into a specific three-dimensional structure. And so that folded shape is so important in terms of the function of that protein that biochemists actually have these different levels of protein folding or different levels of protein structure. And so I wanted to walk you through these quickly, but they're called the primary structure, secondary, tertiary, or quaternary structure of proteins. So what do these terms mean? Well, the primary structure of a protein is the sequence of the amino acids in that polypeptide chain. So just, if we were just to go down the chain from one end to the next, what are the amino acids that are found in that protein? Remember, you have 20 different amino acids that can be used to build the protein. So what is the particular order of those amino acids? And really, it's going to be that primary structure is in effect going to determine the secondary and the tertiary structure as well. So really, this you could argue, initially this is the most important. Uh, so here's what that primary structure would look like. So each box here is an amino acid, and they're just linked together with those peptide bonds shown in red here. So what are these amino acids? And let's say it's histidine, lysine, arginine, whatever the case might be, right? That specific order is going to be your primary structure. And then we take that straight unbranched polypeptide chain and we can start to add a little bit of folding by forming bonds between some of the R groups on those different amino acids. And usually the first bonds that are going to get formed are going to be hydrogen bonds. And there's two common structures that will usually form with this sort of first order folding driven by hydrogen bonds. This is considered the secondary structure. So these two common secondary structures are what are called alpha helices and beta pleated sheets. And you can see examples of both of these down here. But if you look, the little red dots in these images are these hydrogen bonds that are forming between amino acids in a single polypeptide chain. Right? So you'll see one or both of these types of secondary structures forming in basically every protein. And I'll show you an example in a minute, but you could have one protein, one polypeptide chain that'll form, two alpha helices and one beta pleated sheet, for example. Just going to vary protein to protein. But those are common secondary structures. And then we're going to add in additional bonds forming between amino acids. So basically all the different types of bonds that we talked about are coming into play here. Ionic bonds and van der Waals interactions and disulfide bridges, which are bonds between the sulfhydryl functional groups that I mentioned earlier. But once all this bond formation is done, if we were to kind of step back and take a look at, okay, what does this folded protein look like now? That would be considered the tertiary structure, that kind of overall three-dimensional shape of the final folded polypeptide. So you can see in this particular example here, right, this is just one polypeptide chain, right? Here's one end of the chain. Here's the other end over here. You can see we formed like two alpha helices, one beta pleated sheet. There's some other bonds going on here. We've got a hydrogen bond over here. We've got a disulfide bridge here, right? So in this case, the sort of the final shape we ended up with is almost like a triangular tertiary structure. But again, it's going to be different for each protein, right? Depending on what amino acids it contains and what its R groups look like. we may form more or less of these different types of bonds and end up with different shapes of proteins in the end. But again, it's really that three-dimensional shape that is so important for the function. There's different ways of representing these tertiary structures. We could use the ribbon models, which is kind of what we were just looking at on the previous slide, where the polypeptide chain is a ribbon. Stick models, space filling models, but just different ways to show the same thing. And you'll see all of these as you go throughout the course and the textbook. So that was tertiary structure. Now some proteins, not all, but some have what's called quaternary structure. And this is unique to proteins that are made up of multiple polypeptide subunits. This is somewhat unusual, but kind of the classic example of this would be hemoglobin, which is the protein that makes up your red blood cells, right? Carries oxygen through your bloodstream. Hemoglobin has a quaternary structure because that one functional protein, hemoglobin protein, is actually formed from, in this case, 4 different folded polypeptide subunits that all have to come together. So you can see the 4 subunits down here, color coded, right? Each one folded up into its own 3D shape, but we have to bring all of those together. So that would be quaternary structure. Not all proteins have it, but it's pretty cool if they do. All right, so hopefully you've gotten the idea, right? It's the shape of the protein, the folded shape, that is just critical for function. So the shape is going to be key. Also, what R groups are exposed on the surface of that folded protein are going to be very important, because that'll really help determine the biochemistry of that particular protein, right? What other types of molecules can it interact with in a cell? And I did want to point out that the folding of proteins can be affected by a few different environmental factors. So temperature, pH, really high or low concentrations of either polar or nonpolar molecules can basically interfere with some of these bonds that are being used to fold that protein up and hold it together. And you can quote unquote denature a protein, which is basically unfold it. And as soon as you unfold it, it's not going to function properly anymore. So, for example, this is the reason why a very high body temperature is dangerous and can kill you. If your body temperature gets too high, the bonds that are holding that protein in its folded 3D shape are going to start breaking inside your cells. The protein denatures, can no longer carry out whatever job it was intended to do, and your cell stops working and dies. So protein folding is so important. And it is in fact so important there are actually other proteins whose sole job is to help other proteins fold correctly. And these are called chaperone proteins. These chaperones can literally form what looks like almost a little Tupperware container that unfolded or denatured proteins could enter. So we put in our unfolded polypeptide chain, pop the lid on this chaperone protein complex, and the idea is now that that unfolded polypeptide chain is protected from some inhospitable conditions, weird pH maybe inside the cell, or weird concentration of polar molecules or something. It's protected in there. These secondary bonds are allowed to form and the protein folds up, and hopefully the idea is that we've formed enough of these bonds that it's stable once it's released from the chaperone container. Okay, so that is proteins in a nutshell. We got basic, structure, function. So let's move on to carbohydrates here, which are, of course, carbohydrates are sugars. They are a very important form of both stored and transported energy in pretty much all organisms. Um, they can also be used quite frequently just as sort of carbon backbones for building lots of other molecules inside of cells. I mentioned in the first section that carbohydrates are also polymers, and the monomers we use to build carbohydrates are monosaccharides, aka simple sugars. If you link two monosaccharides together, you form what's called a disaccharide, di meaning two. You link a few more together, you get what's considered an oligosaccharide. But really here we're going to be focused on polysaccharides, which are going to be just hundreds or thousands of monosaccharides linked together. We really have these three arguably most important, biologically important polysaccharides, which are starch, glycogen, and cellulose. We'll talk a little bit about those in a minute. But all three of those polysaccharides, starch, glycogen, cellulose, are all polymers of a monosaccharide called glucose. And glucose is really a preferred energy source in many cells, so this is one that you'll see quite often. Glucose is a 6-carbon monosaccharide, 6-carbon sugar. It can exist in this straight chain form, but more often than not in the cellular, aqueous cellular environment, it is going to circularize, so you'll end up with something that looks like this, circular form of glucose. I don't want to get too hung up on this, but you can have what's called alpha or beta glucose, which really just comes down to the orientation of this one little hydroxyl group here. Is it pointing up or down? But you'll see that nomenclature if you read this chapter, so just so you're aware of what that's referring to. And although glucose is really going to be the most important monosaccharide for us, I did want to point out that there are lots of other monosaccharides out there. They always tend to get categorized based off of how many carbons that particular monosaccharide has. So like I said, we were just talking about glucose, which was a 6-carbon monosaccharide. That would make it a hexose. There's other important hexose monosaccharides, fructose, for example. We've got 5-carbon monosaccharides, ribose and deoxyribose. We're going to bump into those here in the near future because those are important components of DNA and RNA. You can even have a little 3-carbon glyceraldehyde monosaccharides. But for our purposes, we're going to primarily focus on glucose, as I said. So we need to take these monosaccharides and start linking them together if we're going to build a polymer. And so we do that again with our dehydration condensation reactions, right? We remove a water molecule, and then we are left sharing electrons between these two monomers to form a covalent bond. And just like we saw in proteins, there's a special name for the covalent bond here, and it's called a glycosidic bond or a glycosidic linkage. Remember, in proteins, it was a peptide bond, peptide linkage, but it's just a fancy name for this particular covalent bond, just two atoms sharing electrons. All right, so we link our monosaccharides together to form these really long, giant polysaccharides. And I wanted to focus on the three big ones in terms of biology, which, as I said, were starch, which is a storage polysaccharide in plants, right? For example, a potato tuber is just that potato plant storing away as much starch as it can in that below ground structure. Glycogen is the storage form of sugar in animals. And then you have cellulose, which is a structural polysaccharide, of course, found in the cell walls of plants. And what you can see here are the differing molecular structures of cellulose, starch, and glycogen. And you don't have to memorize these, but just wanted to point out cellulose is just a straight chain polymer of glucose. So glucose linked to glucose linked to glucose form this long straight polysaccharide. And what you'll find in the cell walls of plants, right, where cellulose is really predominant, is you'll get just lots of these cellulose chains kind of bundled together, interwoven with each other, just to provide this dense, protective structural covering around plant cells. In terms of starch and glycogen, which remember those stored forms of polysaccharides in plants and animals respectively, you'll notice these are branched. So we have side branches coming off, but again, it's just glucose linked to glucose linked to glucose. And really the main difference between starch and glycogen is just how dense the branching is. So you can see in starch, the branches are a little bit more spaced out, a little farther apart. In glycogen, it's very dense branching. All right, so for our purposes in this class, we're mainly going to focus on those three key polysaccharides. But I did just want to mention, there are some other biologically important carbohydrates, of course. So I just wanted to run through a few examples here. We've got what are called sugar phosphates, amino sugars, and chitin, just to show you a little bit of diversity here. So sugar phosphates are so named because they're a sugar that has a couple of phosphate groups attached. Remember, that was one of those functional groups we were talking about. And sugar phosphates you see in some of these key energy reactions that are occurring in cells. And in fact, this particular example here, fructose 1,6-bisphosphate, we're going to see again in an upcoming chapter when we look at cellular respiration. This is a common intermediate found during that process. But again, you might remember the phosphate groups are typically found in energy-rich molecules, so that shouldn't be a big surprise to you that this is a key energy intermediate. Here we've got the amino sugars, which again are aptly named because these are sugars that have an amino functional group attached to them. And amino sugars are really important components of cartilage. So you might have heard of glucosamine. Somebody has arthritis, which is a deterioration or reduction in the amount of cartilage in their joints. They'll often take glucosamine supplements, try to replenish some of the missing amino sugar. And then the last one to mention here, chitin, has this big complex structure. But this is another example of a structural polysaccharide, like cellulose, for example. But this is the one that makes up the exoskeleton of insects. So if you ever see the little cicada shells that you find on the trees in the summer around the Midwest, those are pretty much pure chitin. And if you've ever picked one of those up, it's pretty tough and fibrous. And in fact, it's such a strong, flexible polysaccharide. Chitin is even used in surgical threads in some cases. All right, so I'm going to stop there, and we will pick up the last section of Chapter 3, which is going to be about lipids, in the next part.